Hello, today I'm going to talk about overlaps and distinctions between long COVID and ME/CFS. I'm at the Department of Molecular Biology and Genetics at Cornell, and in addition to my position in that department, I'm also director of the Center for Innervating Neuroimmune Disease. This center is entirely devoted to the study of ME/CFS. We have 3 departments at Cornell involved, as well as the Hospital for Special Surgery. I want to start by telling you a little bit about myalgic encephalomyelitis, also known as chronic fatigue syndrome. Myalgic means pain or tenderness in one or more muscles, and encephalomyelitis is an inflammation of the brain and spinal cord, typically due to an acute viral infection. This name was coined by an MD in London in 1955 who was involved in treatment of patients who became ill during an outbreak. Later, in 1988, the CDC recommended the name chronic fatigue syndrome. The reason that was claimed for this change in name was that there wasn't actually proof of encephalomyelitis, although now there is actually some indication that there is encephalomyelitis in ME/CFS. The patients who have ME/CFS detest the name chronic fatigue syndrome. Hence, ME/CFS is used as a compromised name. The diagnosis of ME/CFS is made by several different criteria. The one I'm going to highlight is the criteria recommended by the Institute of Medicine in 2015. A committee met for a whole year to come up with a diagnostic criteria that could be used for the illness. You have to have post-exertional malaise, unrefreshing sleep, and new fatigue for more than 6 months, as well as either cognitive impairment or orthostatic intolerance. The fact is now that some long COVID victims share symptoms with ME/CFS patients, and some of these long COVID cases fulfill the 2015 ME/CFS criteria. In addition to the 5 criteria from 2015, many additional symptoms are shared by individuals with ME/CFS and long COVID, as shown here. Particularly the muscle pain and the night sweats are indicative of an infectious process. There are some long COVID symptoms, though, that are not typical of ME/CFS. For example, people with ME/CFS don't have loss of sense of taste or smell, and they typically do not have hair loss. There may be other symptoms that will be discovered to be more prevalent in long COVID versus ME/CFS. Nevertheless, both long COVID and ME/CFS are very serious disabling illnesses, and there's an estimated 65 to 70 million people suffering from each of these illnesses. Long COVID patients, however, have been ill for 4.5 years at most, but many ME/CFS patients have been ill for 4 decades after worldwide outbreaks that occurred in the 1980s. The most severely ill ME/CFS patients are completely bedbound. Some are tube-fed, not able to speak nor tolerate sound or light, and one such victim is shown here on the right. What we're seeing is that some of the most severe long COVID patients have descended into a similar bad condition. The prognosis for ME/CFS is poor. An estimated 5% recover to normal activity levels, and then unfortunately there's really no effective treatment. We don't know the prognosis for long COVID yet because it's not been long enough to be sure. Some with ME/CFS who are severely ill, ones who are bedbound in the first 5 years of illness, will recover to be better, but perhaps only housebound, or they might even become able to work part-time. There seems to be a transition around 5 years. After that, complete recovery by adults is very rare. About 80% of children and adolescents who became ill during a particular outbreak were studied later. They reported they have recovered as adults, but even these so-called recovered individuals, when they were screened carefully, it could be seen that they did not have normal health. One thing I would like to point out is the issue of calling post-COVID illness ME/CFS if someone fulfills the 2015 IOM committee definition. One issue is that this 2015 committee couldn't know about SARS-CoV-2, If the committee were constituted today, there might be definitions for pre-2020 illness versus post-COVID illness with similar symptoms. And another issue is that some individuals with long COVID who have filled 2015 ME/CFS criteria were hospitalized and have obvious organ damage that individuals who became ill before 2020 do not have. The definitions of PASC or long COVID are currently in flux. And as mentioned, patients with, say, ventilator damage and those with lingering illness are both PASC, even though there's really a difference in what's likely causing their post-acute sequelae. No one who developed ME/CFS before 2020 acquired it from SARS-CoV-2. Therefore, my recommendation is that individuals with PASC who fit 2015 or other ME/CFS criteria could be referred to as having post-COVID illness that fulfills ME/CFS criteria, but not merely ME/CFS, in order to prevent confusion with pre-2020 ME/CFS. We really need to know more about the similarities and differences of the two illnesses before referring to them as the same thing. There are major similarities in onset. Both are caused by viruses. The inciting illness is sometimes mild and possibly asymptomatic. The pre-2020 outbreaks of ME/CFS were likely caused by plus-strand RNA viruses, namely enteroviruses, as I will describe more later. And an enterovirus, like SARS-CoV-2, has caused a pandemic. SARS-CoV-2, like enteroviruses, is a plus-strand RNA virus. So what is the evidence that enteroviruses can cause ME/CFS? In this talk, I don't have time to go into this in detail, but these 5 books I'm showing here, if you read them, I think you'll be convinced that enteroviruses can cause ME/CFS. And I'd also like to highlight something that many people don't realize, and that is that there's been a large number of outbreaks of ME/CFS in the past. Many papers during these outbreaks, earlier outbreaks, used serological methods and detected anti-enteroviral antibodies in ME/CFS patients. In some cases, they didn't detect more people with antibodies in ME/CFS versus controls, and that's one problem about using serological studies. Enteroviral infection is so common that many of your controls will have antibodies to enteroviruses. More useful is using modern molecular methods, especially on tissue. Some of these detected enteroviruses. And as you've probably heard in preceding talks in this meeting, tissue is particularly valuable for searching for SARS-CoV-2. As I mentioned, I don't have time to discuss all of the evidence here, but I did give a talk in the Research Roadmap series that you can access. And also there is a review article written by a graduate student, Adam O'Neill, and myself, that details this information about past studies for infection in ME/CFS in more detail. Another similarity is that both SARS-CoV-2 and certain enteroviruses have been found to persist long after the acute infection is resolved. I think probably a speaker before me has talked about the findings of persistence of SARS-CoV-2. These are just 2 of a number of different papers on SARS-CoV-2. But there are also a large number of papers showing that enteroviruses can persist in tissue. For example, in pancreas and type 1 diabetes, they can persist in cell lines. They've been found in heart and other tissue. Recent example of persistent infection was this study performed in 2008 by John Chia's group. He actually was able to do 165 GI endoscopies and get stomach biopsies, which were then screened for anti-EV with an anti-EV monoclonal antibodies to look for the viral protein VP1. And what he found was that 135 of the CFS samples and 7 of 31 controls were positive for this viral protein. In addition, he was able to identify in some biopsies RNA that was similar to enterovirus sequences. Another similarity between the two illnesses is that an asymptomatic COVID-19 infection can result in long COVID. This is just a headline from New York Times, which pointed out a study that showed that 32% of long COVID cases at that time started with asymptomatic infections. I have a hypothesis that an asymptomatic enteroviral infection could also lead to ME/CFS. The CDC estimates that 10 to 15 million asymptomatic enteroviral infections occur each year. It's a very common illness. A substantial fraction of ME/CFS patients are unaware of any preceding infection. Perhaps about a third do not know how they ended up with ME/CFS. They therefore often describe their onset to a physical or emotional stress, but my suspicion is that the actual cause is probably an asymptomatic infection. The mechanism of the persistence of the SARS-CoV-2 and its viral antigens is unknown, but the mechanism of enteroviral persistence has been well studied in heart, pancreas, brain, and cell line. This is an example of something from ME/CFS that could be instructive for studying long COVID. In enteroviral persistence, there is evidence for steady state infections in which many cells are infected, but there's a low viral replication that results in a non-lytic phenotype. So it's known in enteroviruses that steady state infections can result from defective viruses. There's a sequence at the 5' end of enteroviruses. Here's a diagram of one here that is important for replication. And 5' terminal deletions of this sequence affect replication. 5' terminal deletions have been seen in tissue in myocarditis and cardiomyopathy, and they have also been seen in samples from ME patients. Something similar may be happening in the case of SARS-CoV-2. One difference between enteroviruses and coronaviruses is that enteroviruses lack a proofreading mechanism, while coronavirus genomes actually do undergo proofreading. Nevertheless, a lot of different variants have been identified in coronaviruses. Long-term infection of immunocompromised individuals by either type of virus can give rise to new variants, which have, as we well know, have very different characteristics. One thing that occurs in both long COVID and ME/CFS is reactivation of herpes viruses. These are just a few papers demonstrating the possible involvement of herpes virus infections in both long COVID and in ME/CFS. It was known as long ago as 1992 that after an outbreak of ME/CFS, that there was reactivation of HHV6, for example, here. And a number of patients will tell you that they developed their ME/CFS after mononucleosis. So herpes viruses like EBV may be accomplices in both long COVID and ME/CFS. So an active herpes virus infection could be making an individual more susceptible to another virus, but it's also possible that continued expression of herpes virus proteins could be contributing to pathophysiology, and this clearly needs to be studied in more detail. Another similarity between long COVID and ME/CFS is that there are characteristics of T-cell exhaustion when immune cells from both types of patients are analyzed. Let me just mention the NCI definition of T-cell exhaustion. It occurs when T cells lose their ability to kill cancer cells or cells infected with a virus as a result of long-term activation of the immune system. These exhausted T cells have high amounts of immune checkpoint proteins on their surface, which can suppress their activity. Whether or not T-cell exhaustion is a major factor in long COVID is under study, and perhaps some preceding talks mentioned this. Here are just a few papers that are beginning to be reporting evidence for T-cell exhaustion in long COVID. In the case of ME/CFS, I don't have time to go through in detail through each study, 3 of which have been done associated with my lab. So I'm just going to summarize what we know now about possible T-cell exhaustion in ME/CFS. This diagram of an exhausted T-cell came from a review, but I've shown on here what we know about T cell exhaustion phenotype of immune cells in ME/CFS. We know that glycolysis is lower in ME/CFS T cells, and fatty acid oxidation is increased. Those are characteristic of exhausted cells. In addition, GLUT1, glucose transporter, is increased, and the inhibitory receptors, PD-1 and SLAMF6, are increased in ME/CFS. Furthermore, the production of interferon gamma and TNF-alpha is lower in ME/CFS immune cells than in normal cells. So this is all evidence that there could be exhaustion occurring in ME/CFS. More information about this can be seen in a talk I gave in an April Portugal ME/CFS meeting. So patient symptoms in both ME/CFS are diverse and can be clustered into groups. A postdoc in my lab, Catherine Glass, identified 4 clusters that differed between whether they had low severity and low key symptoms versus high severity and high symptoms. So there were 4 clusters in this small ME/CFS group of patients that were analyzed. A much larger study was done with the RECOVER cohort, and again, 4 different clusters were found in the long COVID patients, clusters of symptoms. We don't know if these 4 clusters correspond to the ones in ME/CFS because we use different symptoms to make the clusters and because we had different data. But one thing that's important about this PASC study is that a single virus type can give rise to a post-viral illness that has diverse symptoms in different people. And it means also that pre-2020 ME/CFS may have also been caused by a single virus type rather than multiple viruses. A number of people have invoked multiple viruses for the reason of ME/CFS subgroups. But what we know from long COVID, that a single virus can cause these different clusters. So what are some of the differences between long COVID and ME/CFS? They aren't incited by the same virus. The virus leading to long COVID is more deadly. During the outbreaks of ME/CFS, we did not have the death rate that we had in the outbreaks, initial outbreaks of COVID-19. The virus causing long COVID mutates less often than enteroviruses, and some long COVID symptoms are not seen in ME/CFS. But what else? Well, one thing is that there's lower levels of hypercoagulability in microclots in ME/CFS versus long COVID. You've probably already heard in this meeting something about the problems with hypercoagulability. Low levels of cortisol in long COVID were reported, but we did not observe these in ME/CFS. So in an earlier study that we performed in 2022, the cortisol levels between patients and controls were very similar. Now, we will have to consider that these 2 studies weren't done at the same time and potentially with different technology. So this is something that could be investigated by having the same lab look at cortisol in both groups of patients. The same is true for studies of serotonin. In the case of serotonin, recovered patients had higher levels than individuals with long COVID, but we did not see a difference in serotonin in our earlier study. I would like to suggest that we need to be careful about confirmation bias. There's an unwarranted assumption that because long COVID and ME/CFS share most of their symptoms, the biological differences between people with the 2 diseases versus people who are healthy must be the same. We don't actually know if that's true. We need to find it out. We need to study this and not just assume they're going to be the same. Now, here's an example of a study that says here, it's a preprint saying that oxidative stress is a shared characteristic. Well, that's true. If you look at this red and green, the red and green here, that's the level of oxidative stress in these 3 types of cells in ME/CFS and long COVID. But what's being ignored in this study is the fact that the people with the cells from people with ME/CFS have much higher levels of oxidative stress than the cells from people with long COVID. Likewise, here's an example of a pilot study on the proteome, a plasma proteome, in which the conclusion was that there's changes in pathways similar in ME/CFS and long COVID. But then if you look at the data, you see that actually there were only 9 proteins that were upregulated in both diseases. And actually, most of the proteins between long COVID and ME/CFS that were upregulated were different. And the same is true of the downregulated proteins. So I would urge people to not just assume the molecular level, the differences at the molecular level are going to be the same, but instead we need to find out how similar they are and not sweep differences under the rug. So the conclusions I would make are that long COVID and ME/CFS share many but not all symptoms, that both are post-acute viral illnesses. Individuals who something that needs to now be remembered, that individuals with new onset of the identical long COVID and ME/CFS symptoms could have been infected either with SARS-CoV-2 or with pre-2020 ME/CFS viruses. So what I mean by this, at least some current so-called long COVID cases might not actually result from COVID-19, especially if the case was asymptomatic and there's not good evidence that it started following it immediately after a COVID-19 infection. Both SARS-CoV-2 and enteroviruses can persist following the acute infection. We don't know the role of virus persistent and herpes reactivation in long COVID and ME/CFS, and this needs further study. T-cell exhaustion may be involved in both. And what's interesting is that T-cell exhaustion can be caused by chronic viral infection, which may be occurring in both. Right now, a few molecular differences between long COVID and ME/CFS have been observed, but we urgently need research to determine whether molecular markers can distinguish these two illnesses in order to ensure appropriate treatment. An example of this is that treatment with drugs that are specific to SARS-CoV-2 are not going to help people who have ME/CFS who became ill before 2020. I'd like to end with some acknowledgments of people in my lab and many collaborators who've contributed to this study. Thank you.